Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 9th July 2020. In our previous session we talked about epidermal tissue, types of epidermal tissue, functions of it, location of it, and in this session we are going to talk about muscular tissue. Muscular tissue is responsible for formation of muscles. So 40% of mammals' body weight is because of this tissue, muscular tissue, and it originates from mesoderm. Remember we said three layers are responsible. One is ectoderm, second mesoderm, third endoderm. So mesoderm forms muscular tissue. And what are the properties? Muscular tissue constitutes the muscles and made up of myocytes. A muscle cell is called myocyte, M-Y-O-C-Y-T-E-S. So muscle cells are myocytes, and these are in the form of contractile fibers, varying in length, present in cytoplasm, known as sarcoplasm. In cell we say cytoplasm. In muscle cell, that is in myocyte, the liquid that is present is called sarcoplasm. So it develops from the mesoderm, as I said just now, of the embryo. But the muscles of the iris of the eye and myoepithelial cells of salivary, mammary and sweat glands arise from ectoderm. These are exceptions. The special property of muscular tissue is contractility. Contractility, that is, the cells of the muscle tissue can shorten, they become short, and on return original relaxed state, so they can become constricted and relaxed. The muscle cells contract in a definite direction. This is the best property of the muscular tissue, contractility. And the second is electrical excitability. It is due to the energy stored in the electrical potential difference across the plasma membrane. So, general structure, if you see, the muscle tissue consists of long, narrow cells called muscle fibers. These muscle fibers are made up of parallelly arranged proteins called myofibrils, and just now I said the cytoplasm of the muscle cell is called sarcoplasm, and the cell membrane is called sarcolemma, and the endoplasmic reticulum of the muscle cell is sarcoplasmic reticulum, and mitochondria of the muscle cell is sarcosome. Muscle fibers may contain one or more nuclei, and mitochondria are present between myofibrils. So remember, myocytes, muscle cells are called myocytes, and based on the structure, muscle tissue is differentiated into three types. Striated muscles, we also call it as skeletal muscles. Unstriated muscles or smooth muscles. And third, cardiac muscle. Striated muscles or skeletal muscle, or we sometimes call it as rough muscles, and these are voluntary muscles. Whenever we want, we can make them contract and relax. Location, where do you see them? Forms more than 80% of the body mass. It is found in body walls, tongue, hands, legs, that's limbs. And structure, look at the structure. It consists of alternative bands, dark band, light band. And the fibrils, myofibrils, you can see are many. So thick myosin filaments and thin actin filaments. So you got actin and myosin. These are the proteins present in the muscles. And look at that. A sarcomere will be having bands. We call it I band, A band, and I band. And you got internally discs. These are called Z discs. And you got a thick area that's called H zone and M line. So dark bands, these are anisotropic, refract light differently in different planes. and are called anisotropic or A band. That's A band. That's what we said, A band sarcomere. Its middle part is light and termed Henson's line or H-zone, Henson's line. What about light bands? They are isotropic, have some refractive index in all the planes and are called isotropic or I band. It has a membrane in its mid called membrane of cross or Z lane or Z band. It's zigzag in structure. How do you differentiate A band and I band? A band, they are anisotropic. I band, isotropic. Give dark appearance in microscope, give light appearance in under microscope. Bisected by H zone, as you are seeing in the diagram, and bisected by Z band. Length remains unchanged during muscle contraction, whereas I band, it shortens during muscle contraction. And sarcomere is a part of myofibril between two successive Z bands, sarcomere, because flesh part. It's defined to be called the functional and structural unit of muscle fiber, and it includes all the bands. It's around 2 to 3 UM long. It contains a hemoglobin-like pigment called myoglobin. See, in blood we have got hemoglobin, in muscle we have got myoglobin, which can take up, store and give up oxygen according to the requirement. Under electron microscope, if you see the muscle fiber, you can see two types of myofilaments. Primary myofilament, which is very thick, and secondary myofilament, which is thin. And as I said, two proteins are responsible: actin, thin filament; myosin, thick filament. So they are confined to A band only. Primary myofilaments are confined to A band. They are made up of protein myosin. You must remember this word myosin for the exam sake. They bear minute projections called cross bridges. Look at the diagram, cross bridges of the protein. They are free at both the ends. Whereas secondary myofilament, which is thick, they are confined to I band but also extend towards A band, making an overlapping. So this overlapping occurs between primary and secondary myofilament, and overlapping occurs in A band, providing dark appearance. So these are made up of three proteins. Mainly it is myosin. Along with that, actin, tropomyosin, troponin. They are attached from one end at a Z line, while another end is free. It gives Z line a zigzag appearance. Now, general feature. The striated muscles, also known as voluntary muscles, skeletal muscles, rough muscles, because of its contraction, is in our control and helps in locomotion, movement. And unstriated muscle or smooth muscle, look at the diagram. There's a plasma membrane, myofibrils are there, nucleus is there, sarcoplasm is there. Where do you see them, these smooth muscles? They are present in hollow internal organs called viscera, such as alimentary canal, genital tract, hence they are also called visceral muscle. It also forms dermis of the skin. What is the function? They are responsible for involuntary. See, first one is voluntary, whereas this is involuntary activities, and are often called involuntary muscles. So, structure, they don't contain any strips. They are smooth muscles. There are no cross walls. Cells are spindle shaped, and one muscle fiber of smooth muscle is smaller than that of the striated muscle. And nucleus is oval, as you are seeing here, and its center. Cells are uninucleated, they are more than one. And myofibrils are hardly to be seen. Function controls involuntary activities. All the involuntary activities are because of this unstriated muscle. The muscle fibers are non-striated, spindle shaped. Each fiber contains a single oval nucleus surrounded by sarcoplasm. In the cytoplasm, the myofibrils are arranged longitudinally. There is no sarcolemma. Okay, and now let's move to the third one, cardiac muscle. Very, very important muscle. Cardium means heart. The muscle that is present inside our heart. And you quickly recollect and realize and reflect that this is the muscle for which we are very, very thankful. the day it has taken birth, the day we have taken birth, from the day we have taken birth until we die, this muscle will be acting and acting and acting. So, if someone says, why don't you take rest? Ask your cardiac muscle whether it is taking a rest, and if it takes rest, what happens. So, location, walls of the heart, superior vena cava and pulmonary vein. Structure. Cardiac muscle consists of short cylindrical fibers which are interconnected by oblique bridges. So, it's a combination of, we can say, smooth muscle and rough muscle. The cardiac fibers contain large number of mitochondria, naturally because continuously it's working, and glycogen granules, so as to fulfill their high energy requirements for rhythmic contraction of cardiac muscles, heart. They have poorly developed striations. The cardiac fibers are about long, wide. These are uninucleated again, and nucleus is present in the center. What is the general feature? Wave of excitation is self-generated. They are self-generated waves. Blood supply is abundant. Capillaries penetrate the fibers. Lymphatic capillaries are also present. They have striations, portion of it. The cardiac muscle is innervated both by brain, central nervous system, as well as autonomous nervous system. Unlike other two muscles, they form 3D network. So, cardiac muscle is composed of smaller interconnected cells with a single nucleus which appear as dark line, and it's called intercalated discs. Intercalated disc. The intercalated disc function is as boosters of contraction wave and permit the wave of muscle contraction to be transmitted from one cardiac fiber to another. The myofibrils have transverse, faint, dark and light bands, as you are seeing in the diagram. Dark ones and light bands, alternatively they are present. And these fibers have some lateral branches known as oblique bridges to form a 3D network, contractile network. Wall of the heart and in the wall of large veins like pulmonary veins, superior vena cava, these are present. Nerve supply from brain and autonomous nervous system, rapid contraction, never get fatigue, and they are involuntary. And they are involved in contraction and relaxation of heart and large veins. And that is the reason why the cardiac muscle has got a lot of significance. Hence, what you have to do now is draw the diagram of all the three. And I need to tell you that yesterday CBSE has announced some reduction in your syllabus because of COVID-19. And in this chapter, I must tell you that it is only tissues. In structural organization in animals, 7th chapter, already I have put up in our WhatsApp group. Please go through that, open that file. You will see chapter number 7, the deleted portion is morphology, anatomy and functions of different systems like digestive, circulatory, respiratory, nervous, reproductive, insect, cockroach. This is deleted. So only muscles, muscle tissues. Rather 4 types of tissues are there in this chapter. So in structural organization, we are going to study only animal tissues. Also, I need to tell you that the marks pattern also changed. In the previous session, if you recollect from your memory, there are 5 units, and in the 5 units, diversity of living organisms, there were 7 marks. Now, for your batch, it is 15 marks. Okay, and for second, structural organism, plants and animals, present unit, it is reduced from 12 marks. It is only 8 marks. And third unit, cell structure and functions, 15 it was, and it is 15. No change in third unit. And fourth unit, plant physiology, it was 18 marks. For your batch, it is going to be 15 marks. And human physiology, it was 18, now reduced 1 mark, 17. Please go through that accordingly.